So Vibrio is a genus of bacteria. They are gram-negative. And as a lot of these gastrointestinal pathogens are, they're facultative anaerobes, so they can grow in anaerobic environments. They do prefer oxygen. And they're oxidase positive. Remember, with the Enterobacteriaceae, that whole group of gastrointestinal pathogens and friends like plague, they are oxidase negative. So this is a gastrointestinal gram-negative pathogen, but it is oxidase positive. They do a good amount of fermentation. So they do have fermentive metabolism. They are motile, generally through a single polar flagellum, whereas the Enterobacteriaceae, those that are motile, usually have peritricious flagella. They have flagella all over the cell. Most often we see Vibrio in water. Often what we call brackish water, so kind of where the salt water meets fresh water. So estuaries, coastlines where rivers come in. And I'll tell you about like some local, not local to us, but like local US areas where you can find Vibrio. And we classify Vibrio infections. There are two major types of infection that various species can cause. Generally, we see either GI disease, so gastrointestinal infection, or wound infection. Some species are capable of doing both, but generally a species will be associated with one versus the other. When you look at them under the microscope, we'll see it on the next slide, they kind of have a comma shape. They're not a straight rod, they're kind of curved like a comma. The pathogen that's most associated with gastrointestinal disease is Vibrio cholerae. It causes cholera. So it's we'll learn more about cholera in just a minute. Sorry, we're gonna talk about diarrhea again today. We're in the diarrhea pathogens right now. So and then we'll leave diarrhea and then we'll come back to diarrhea. It's always diarrhea. Then there's Vibrio parahemolyticus, which generally is associated with gastrointestinal infection, but can cause wound infection. And Vibrio vulnificus, which is highly associated with necrotizing fasciitis, or like flesh-eating infections. So it's a very dangerous wound pathogen. It can sometimes cause gastrointestinal complications as well. So I told you they're curved rods. They're sort of comma-shaped. They have that polar flagellum. They have a decently wide temperature range for growth, anywhere from 14 to 40. But they do require salt, which is why we typically see them either, along coastlines, in brackish water. They're associated with marine environments mostly. They do tolerate, I would say, it's a decent range of pH. Remember, every number we go up on the pH scale, like when we go from 6 to 7, or if we go backwards, right, 9 to 8, that's 8 is 10 times more acidic than 9. 7 is 100 times more acidic than 9. So it's a decent range of pH that it will tolerate. But notice I said especially Vibrio cholerae, which causes cholera, a gastrointestinal infection. These are really susceptible to stomach acid. They will get destroyed by stomach acid. So the thing to think about then is, how do people get these infections? How do people get cholera if the bacteria is so sensitive to stomach acid? And there are a couple of things that we can talk about that can make it easier to get cholera. One is malnutrition. People who are malnourished tend to have a higher stomach pH. It's not as acidic. So it's more likely that bacteria can make it through the stomach. If the bacteria, any of the Vibrio species, are consumed with a meal, that can temporarily raise the pH of the stomach and allow the bacteria to survive. So we sometimes see some of these Vibrio species associated with the consumption of contaminated shellfish. They basically are able to get through the stomach fast enough because the pH raises when you eat, that they don't get destroyed. So those are a couple of ways they can still cause infection. And of course, if they're going to cause a wound infection, wounds are not acidic, so wound infection is pretty easy. But gastrointestinal infection, often you're looking at people who are malnourished or people who have gotten infected while consuming a meal. Again, the majority of species have a polar flagellum, allows them to be motile. They have pili to help with attachment. Specifically in Vibrio cholerae, they have a pilus called the toxin co-regulated pilus. So that also tells you they have a toxin. We'll talk more about cholera's toxin in a little bit. So they're able to move, and when they get in, they're able to adhere pretty well. Again, this is that chart of the Vibrio species that are most commonly associated or most commonly implicated in infection. There are others. So you may, like if you go to the CDC website and look at Vibrio infection in the US, there are other species of Vibrio that cause infection. These are maybe most dangerous, kind of the type species for these types of infections. So Vibrio cholera from mostly contaminated water or food, and it causes gastroenteritis most commonly. It can cause bacteremia, but that's less common. Vibrio parahemolyticus, especially contaminated shellfish or seawater, causes gastroenteritis. It can also cause wound infection. And then Vibrio vulnificus is the one that's most highly associated with those really dangerous wound infections. Because they're gram-negative, they have LPS, just like all of our gram-negatives. And if you remember when we talked a little bit about E. coli on Monday, we talked about how we can use the O antigen to be part of the strain of E. coli, like O157H7. We use the O antigen to give Vibrio its serogroups too. So we would talk about Vibrio cholera O1 or O139. Those are the ones most commonly associated with causing pandemics. So we can use that O antigen to classify the different Vibrio species into specific serogroups, and they all cause disease a little bit differently. The majority of Vibrio have a polysaccharide capsule except for Vibrio cholera strain O1 or serogroup O1. And kind of unique among the bacteria, Vibrio species have 2 chromosomes. So when we teach a class like microbiology, we tell you, oh, bacteria, they have 1 chromosome. For the most part. Vibrio is just one of those groups. It's a genus that has 2. And there are some strains that also carry plasmids that can confer antimicrobial resistance. Okay. So again, the LPS can cause toxin-like symptoms, right? Toxic shock, things like that. Capsules are really helpful for immune evasion. The capsule can also help protect sometimes from the stomach acid a little bit. But we can get into more specific virulence factors that some of the species have. I won't expect you to remember all of them, but I will highlight what I think are some of the most important. For Vibrio cholera, the most important virulence factor that is going to be the one that causes the disease, cholera, is cholera toxin. Not every strain of Vibrio cholerae has cholera toxin. But the ones that do have cholera toxin, the toxin works by causing an ion imbalance. It causes your cells to hypersecrete fluids. And so when you have hypersecretion of fluids in the GI tract, you have diarrhea. But cholera diarrhea is different from other diarrhea. Cholera diarrhea is like liters of diarrhea a day, like 15 liters of diarrhea a day. So people who get cholera die of dehydration within 2 to 3 days. If they survive that, they usually actually clear the infection on their own, self-limiting. You just have to survive that severe level of dehydration. So when I say diarrhea, it's diarrhea. So that's like the major toxin that some strains of Vibrio cholera produce to cause diarrhea. There are other toxins produced by the cholera bacterium. They have other toxins that can cause diarrhea. So if you are infected with a strain of Vibrio cholerae that does not have cholera toxin, you can still get diarrhea, but it's more of a classic type of diarrhea. It's not like gallons of fluid coming out of your body a day. It's just regular diarrhea. Still unpleasant, but much less dangerous. Vibrio parahemolyticus, it also causes gastrointestinal infection. It has an enterotoxin that can lead to watery diarrhea. Vibrio vulnificus, that's the one that specifically is highly implicated in really dangerous wound infections. It has a capsule that helps it be anti-phagocytic, but it also has a number of different enzymes and proteins that destroy tissue. So it just has a lot of tissue destruction capability. Okay, so the cholera toxin is also an A-B toxin, just like the diphtheria toxin, where you have multiple domains. It's like a subunit toxin. The A subunit is the active domain. So whenever we talk about an A-B toxin, the A does the action inside of the host cell. The B is what binds to the host cell. So the cholera toxin has a single A and 5 B domains. So it has 5 proteins that help it to bind to the surface of the cell.
What B is?
B is binding to the host cell. What the A subunit does molecularly inside of the cell is lead to increased formation of cyclic AMP. It causes an ion imbalance, and that causes the release of water and electrolytes. It's just pure fluid secretion coming out of your cells. So electrolytes, things like sodium, chloride, like different ions, just come out of your cells with the water. Now, without adhesion, you don't get infection. So you either have to have that toxin co-regulated pilus. So the strains that produce cholera toxin will produce the toxin co-regulated pilus, or other adhesion proteins. So it can't just like pass through your GI tract and make you sick. It has to bind and adhere. So if there is no adherence, there is no infection. But like I mentioned before, strains of Vibrio cholera that do not have cholera toxin, as long as they can bind, they still can cause disease. They have other toxins that can cause diarrhea. It's just not as severe. And the only strains that we know of Vibrio cholera that produce cholera toxin are strain 01 and 0139. There is currently an ongoing, winding down pandemic of cholera, and it is a version of the 01 strain. They call it L-Tor. Okay, so I mentioned because of the salt requirements in environmental areas, we tend to see them in marine environments and estuaries, anywhere where you have brackish or salty water. And they flourish, they do even better when the water is a little bit warm. So we don't often hear of Vibrio infections out of San Francisco because it's freaking cold, right? Our ocean is not very warm. Try the Gulf of Mexico. That's really nice for people and for Vibrio. Yes?
So since most Vibrio cause fluid secretion, is that because it survives in high ion environments, high ionic environments, or is it to neutralize pH of the GI tract, since that's usually where cholera infects? Or
Are you asking like why does it do the hypersecretion of fluid?
Yeah, is that something to allow it to survive in delivery better? Like why?
Dissemination. It's dissemination. So we don't really talk about it in this class because it's more of the biology of the organism itself rather than its disease-causing capabilities. But the expression of the toxin is often regulated by quorum sensing. And so what they'll do is they'll adhere, and as they grow and replicate, when it gets to a certain number of bacteria, they start to produce the toxin, which allows them to be passed in the fluids that are secreted. So it allows them to be disseminated back into the water, basically. Yeah, I can't even call it feces. I'm going to show you a picture in a little bit. The fluid that comes out of a cholera patient is just chock full of the bacteria. So it's more of a dissemination than anything else. They can also be found in shellfish and other organisms that are like filter feeders. So I would never recommend eating raw or undercooked shellfish, but that's just me. You do what you got to do. They are spread through contaminated food and water. And it takes a lot of microorganisms to cause infection, often because the stomach acid is so efficient at getting rid of the bacteria. But the more bacteria you ingest, even let's say you're not malnourished and maybe you swallowed water at the ocean, the more bacteria that are in there, the more likely it is that some of them could sneak through and not be destroyed by the stomach acid. So it takes the ingestion of a lot of organisms to cause disease. But we usually only see cholera in areas that have poor sanitation. Basically where people bathe, wash their clothing, and get their drinking water from the same source. That's where you most often see cholera. Right? We don't do that. We don't wash our clothes and take a shower, wash our bodies, and then drink that water. Okay? And we definitely don't do that as a community. I don't know what you do in your own home, but we certainly don't have a community area where people do that. So all of the water that we drink has been treated for any potential fecal organisms, so we don't really see cholera in the US. But in other countries where maybe they don't have access to that same infrastructure, that's where we can see it. So with cholera, it's an abrupt onset of watery diarrhea. Can have vomiting, severe dehydration, and this is the important part, hypovolemic shock. You lose all your fluids. You don't have any more fluid. You need fluid. Your blood will thicken up, right? You don't have any fluid anymore. A milder form of diarrheal disease can occur in the toxin-negative strains, the cholera toxin-negative strains. With Vibrio parahemolyticus, I love the term explosive onset of watery diarrhea. I don't know why it has to be explosive onset, but I guess if that gives you the mental picture of what's happening, then there you go. It's a gastrointestinal infection. It's, sorry, but it generally is self-limited. So after a couple of days, you'll be fine. With the wound infection, it's usually if you have an open wound, it doesn't burrow into your skin and cause an infection, but if you have an open wound, it can get in. Vibrio vulnificus, same thing in terms of wound infection. If you have an open wound and you get into contaminated water, the bacteria can get into your body. But Vibrio vulnificus causes a much more severe type of infection than Vibrio parahemolyticus. So this is severe, potentially fatal infections, redness, pain, swelling, tissue necrosis, and septicemia. So it's a type of flesh-eating infection. Okay, so sorry, cholera. This is a bucket of somebody's cholera waste. That's what it looks like. They call it rice water stool. There's no solid material. It's basically water colored with a little bit of bile. That's it. And these are called cholera cots. When patients have cholera, especially if there's an outbreak in an area, each cot is occupied by a patient who will defecate into the bucket. The bucket is measured, and the patient will be given oral rehydration therapy. So it's important to know how much fluid the patient has lost, because that's how much fluid you need to give them back. And this is a child receiving oral rehydration therapy. That's basically it. Antibiotics wouldn't be effective enough to clear the infection before a patient would succumb to the dehydration. So it's really seeking the early treatment and getting hydrated.
With your diarrhea coming out like that,
Uh-huh.
does it clear all of your intestines, even the microflora that grow in there?
It can, yeah. It can clear. It clears out pretty much everything. Yeah, it's a really. I don't know if anybody. Now I kind of want to know. I don't want to do it because yuck. But what's the microbiome like before and after? Yeah, or what's in there besides just cholera? Yeah, I don't want to. I don't want to do it, but I kind of want to know the answer now. Hey, there you go. I know a guy who studies cholera. So if you're interested in grad school, yes.
Some people will take flatworms or those worms for weight loss.
Oh yeah.
Sometimes to clear that infection, you could probably clear with cholera.
Probably, yeah. Ooh, I know people do that too. Ooh, when we talk about tapeworms, we'll talk more. I just saw a really horrible video. I don't remember who sent it to me, but they're not my friend anymore. Of bears with tapeworm, the fish tapeworm.
I just saw that video.
Okay, is it going viral or something? Is that what it is? And it's like all these bears walking around with like multiple tapeworms hanging out. I was like, oh no.
I sent it to my sister this morning.
Okay, well somebody sent it to me and I don't remember who it was, but that's good because they're off my Christmas card list. I was like, what the heck is this? I thought you would like this. Why? Why would I like this?
I was gonna show you after class.
Oh, why would I like that?
Yeah, gross.
It's very gross.
I showed you right before we were gonna eat too. Yeah.
Oh, you showed it to her?
She showed it to me right before we were gonna eat.
Lovely. You're gross, dude. That's nasty. And that's what you get for eating undercooked fish. That's a guy turned up in Fresno with it from undercooked salmon. It's the same tapeworm.
Another reason why I don't like seafood.
Boom. I got you. But so this was back in 2017. There was an outbreak of cholera in Yemen that infected over 500,000 people. Closer to us, after the major earthquake in Haiti, there was a huge cholera outbreak in Haiti. And actually, they think that it was brought by the relief workers. So the relief workers, like the Red Cross, had been in other parts of the world, and they came to Haiti to help, look for survivors of the earthquake, to try to rebuild infrastructure. They had a camp set up, and then downstream of the camp, everybody was getting cholera. So it took hold in Haiti and really affected them because not only, were they like dealing with an earthquake, now they're dealing with cholera on top of that. Kind of the scientist who's considered to be like the father of epidemiology is a guy called John Snow. So they were having a cholera outbreak in London, and he was trying to figure out, well, where was the cholera coming from? And eventually, he found where all these patients were that had cholera, and almost everybody that had cholera had gotten their water from a particular pump. So this was before indoor plumbing. You would go to a well, the closest well to your house, it had a pump. You would pump the water and then take it back to your house. And so all of the homes where people had cholera got their water from this particular well. It's called the Broad Street pump, because it was on Broad Street. He unhooked the handle and the cholera outbreak ended. And it turned out that somebody had dug their chamber pot a little too close to the well. And so the bacteria from the feces had gone into the well. But as soon as they took the pump off the well, everybody who had cholera, unfortunately, either died or got better. Those were your options, and the outbreak ended. So that's kind of the father of epidemiology from cholera. Okay, questions about cholera? Yes.
This is a really random question, but on the cots, do they just like lay there?
Yes. You literally just
all day.
Mhm. I mean, if you think about the last time that you were sick, gastrointestinal sick, you're like, man, I've been to the bathroom I don't know how many times a day. But basically, it's like constantly leaking out of you, like how worn down you're going to be, how tired you're going to be, how dehydrated. They'll basically, just lay there with their tush in the hole. I imagine they get a blanket to like cover the front, and then somebody will come by and take the bucket away.
What if they take it away and then?
Well, it's splash zone. I don't know if it's like constant, but it's like, I mean, some of the bacterial and viral infections can cause people to have diarrhea like 10, 12 times a day. So if you imagine like that many times and it's just pure fluid, you're probably just like, whatever. I don't even care what people see anymore. You just lay there and let them take your bucket. Mm-hmm.
How big do the wound infections have to be? Like, if you just have like a scrape and you go swimming in like one of these places, like, are you gonna get
It's possible.
like just like the smallest scrape?
Yes, any open wound. So this was one from 2017 where he had been swimming off the coast of Galveston, which is outside of Houston in the Gulf of Mexico in Texas. He had a relatively fresh tattoo. And you can, it's kind of hard to see again with the lights and stuff, but you can on the computer maybe see all the swelling and like red, purple. His tattoo got infected and he died of this infection. So I have more headlines, mostly from like Texas, because this is where Corpus Christi, Texas is. So just a little bit south of Houston. A woman dies after contracting Vibrio from swimming in the Gulf of Mexico. Here's another one. You should avoid raw oysters because they are filter feeders. Like, they will have a really high concentration. If the bacteria are in the water in that area, oysters and other shellfish in that area that are filter feeders will often have a really high concentration. And then they're raw, right? You're just like asking for it, basically. Then there was another headline from 2018 of a Vibrio outbreak linked to crab meat imported from Venezuela, where also the water is warm. And so this was from the CDC, Vibrio parahemolyticus and vulnificus. We don't tend to see a lot of cholera in the US because we have decent sanitation. But I'll tell you where you can find cholera in just a minute. But you see, we started to see some increases 2014, 2016. And what, people are talking about is this idea that as global temperatures change, particularly with increases in ocean temperatures because of climate change, we may start to see this bacterium and others spread to areas where we typically did not see them before. And so ongoing, or I guess just this past summer, 6 people off the east coast of the US died of the same infection. So where can you find cholera in the United States? If you were curious and wanted to go look for it, in the Rio Grande. And if you know anything about a little bit of geography of the US, the Rio Grande is the border between the US and Mexico, and I'm talking particularly in South Texas. So when I did my PhD, the lab next door studied cholera, and so they would go down a couple times a year and sample the Rio Grande, and it always had cholera in it. But generally our sanitation prevented people from getting cholera in the US. Okay. So they are sensitive to drying and acidity, so they have to be in that warm, like moist environment. Cultures, if you suspect a cholera infection and you do want to culture it, you need to plate it right away. There is Vibrio-specific media. Again, it's got a little bit of a higher salt content than you would see for classic bacteria growth media. I imagine those of you working in clinical settings in the US, you would really only be looking for not cholera species of Vibrio, so Parahemolyticus, Vulnificus, and other related species. You typically won't be looking for cholera in the US, but you would be looking for the other species. For diarrhea, we typically don't do antibiotic treatment. Your microbiome is already messed up enough. One, because you have all this fluid coming through. Two, you don't want to mess it up with antibiotics, which can make you prone to other gastrointestinal pathogens that can cause diarrhea. But three, it's usually not going to be fast enough. Right? A typical course of antibiotics is anywhere from 10 to 14 days. Even non-cholera diarrhea, 2 or 3 days. It's very self-limiting. Cholera diarrhea, what's most important is fluid replacement therapy. Do not let your patient get too dehydrated. And remember, you wouldn't want to give pure water. They're losing water and electrolytes. So it's an oral rehydration solution that will have electrolytes in it. So you don't, kill your patient with water intoxication. But for the wound infections, something like Vibrio vulnificus, you may, and you should, seek antibiotic treatment. So your patients, commonly you would want to do combined or combination antibiotic therapy because the infections can progress very quickly and some of them can have antibiotic resistance. So you'd want to pick a couple of different drugs. The clinical recommendations will differ like every time I turn around. So I'm not even gonna tell you like what antibiotic to pick because I'm sure it'll be different. But there are, those of you going into clinical lab science, as we get through the semester, we're gonna go through a couple of the different clinical organizations and how they recommend which antibiotics you should pick. So we'll look at those guidelines for picking antibiotics for various infections. So we'll do that in the lab. There is an oral cholera vaccine available in other countries. Sometimes the efficacy is debated, but at the same time, I would probably take it if I lived in one of those areas, just on the chance that it would protect me from getting cholera. Generally, they do an oral vaccine instead of an injected vaccine because that's where cholera causes infection. It's a gastrointestinal infection. So if you can induce something like a robust IgA response, that secretory antibody, that's going to be most beneficial. So that's why even in the US, when children are vaccinated against rotavirus, which is a GI virus, it's actually an oral vaccine because you get the best immune response in the target tissue. So when available, you try to get the best you can. So an oral cholera vaccine.
Let's say somebody had the
Cholera? Mm-hmm.
Yeah, and they were going to the bathroom at their home. Is it super unlikely that somebody in their home would get it as well because of the water?
In their home, I would say it's unlikely only because it's sensitive to being dried out, right? Mm-hmm. So with a lot of gastrointestinal bugs, right, which is not the best term, but whatever, they do go around in households, especially viral infections, because a lot of them, and we'll talk about why, but are less prone to being degraded in the environment. Mm-hmm. So they're not sensitive to being dried out. Somebody with their poopy hands, or you're cleaning the toilet because they're very sick or whatever, you're much more likely to get it. I would think it would be one of those where you could probably take enough precautions that you could protect the rest of the household. It wouldn't be as likely as some of the other GI infections. But, that also depends on the hygiene of the person who's cleaning themselves. Are they cleaning themselves? Are they washing their hands? So it's possible that you could get cholera from someone infected in your house, but it's also possible to protect, I think, in that case. But, if somebody in the house gets norovirus, you're all screwed. That's pretty much where we go. Other questions about Vibrio species? Cholera, Vulnificus, Parahemolyticus. And it makes you paranoid, right? Like now I can't go swimming in the warm water. You go to Cancun, you're gonna get like vulnificus or whatever. Just if you have wounds, try not to go in the water. If you've gotten a fresh tattoo, sorry, wait till it heals. Yes.
Is vulnificus capable of causing uveitis?
As far as I know, I've only seen it associated mostly with wound infections, though there have been some cases that I've seen in the news where they implicate it in GI infection, like if people ate a contaminated food product. But sometimes I question, like I just saw one a couple days ago where they said this woman from San Jose ate some undercooked seafood product and she got a Vibrio vulnificus infection that caused her to lose her limbs. Because when you get a septic infection, right, it can cause you to lose your limbs. And they said because she ate it, like it was a wound infection, and it turned out she actually didn't have Vibrio vulnificus. It was something they thought she might have at first. So all of the articles were like, oh, she had this Vibrio infection, now she lost all her limbs. But it turned out it's probably something else. So it's hard to say whether or not the GI symptoms are Vibrio vulnificus or maybe a different Vibrio species. So I would say it's not impossible that it could cause GI, but parahemolyticus is more associated with GI. Yeah. Yes.
For the GI, is the preferred portal of interest the mouth or?
Yeah. It's going to be like drinking contaminated water or eating contaminated food most frequently. Yeah. But wound infections, it's the same like we talked about with the nail salons. If you shaved your legs and you've got some cuts, the bacteria can get in. Will they do that to everybody who's recently shaved their legs and gone swimming in the Gulf of Mexico? Absolutely not. But if you're unlucky. So it's, if you have a big, giant, fresh tattoo, you don't get to go swimming in the ocean until your tattoo has healed. Yes.
So then how would you differentiate between cholera infection and parahemolyticus infection in the GI tract for cholera that don't produce cholera toxins?
You'd have to grow them up in the lab and do serological testing to differentiate because non-cholera toxin producing cholera strains and Vibrio parahemolyticus both cause kind of a self-limiting diarrhea. They may not go to the trouble to differentiate. They might just say, oh, the person had a Vibrio infection. But you could do it. It would just require like a serological type of testing or genetic testing. Yeah, like certain PCR. Mm-hmm. But in terms of culture, they would look the same. Yeah. Any other? Okay, so we'll introduce a group called the Pseudomonads. These are all gram-negative rod-shaped bacterium. They are generally oxidase positive. They are most commonly associated with skin and soft tissue infections and respiratory infections. Some major genera in this group are listed here. We're not going to talk about all of them. We'll talk about a couple. We worry about especially Acinetobacter and Pseudomonas as hospital-acquired infections that are multi-drug resistant. Burkholderia, we worry about a lot in patients that have cystic fibrosis. So if a patient has cystic fibrosis, the only cure for cystic fibrosis, I say cure because you're not actually fixing the person genetically, but you're fixing the organ that's wrong. The major issue with cystic fibrosis is the buildup of mucus in the lungs. It's a defective ion transporter that leads to mucus buildup in the lungs. And when they have that mucus buildup, they're really, really prone to infection. And Burkholderia tends to be the one that kills most cystic fibrosis patients. The only treatment for cystic fibrosis that provides a lifelong relief from those lung symptoms is a lung transplant. But patients who have Burkholderia cannot get a lung transplant. So even if they are going to die, they cannot get a lung transplant unless they are completely free of Burkholderia. So we worry about that one a lot with our cystic fibrosis patients. We're not really going to talk about Stenotrophomonas either. It's in the same group, closely related. There's it's probably like a 10-year-old documentary now. It's called Hunting the Nightmare Bacteria. And they talk about a Stenotrophomonas infection that went around a hospital setting that was multi-drug resistant. So it's not as common as the others that we will talk about, but it does happen. So I can't talk about everything can't talk about everything. So I try to pick the ones that are most common and you're more likely to see. And that could totally change. Maybe in future iterations of this class, Stenotrophomonas will be like, so I'll have to talk about that one instead. I change it every time. But the first one we'll talk about is Pseudomonas. And the species we'll talk about is Pseudomonas aeruginosa. So those of you who took microbiology here, I know for sure that you have worked with this organism. It's the one that produces a blue pigment on the plate. So they are gram-negative rods. They are pretty much found everywhere. Pseudomonas is a really hardy genus. They will grow pretty much anywhere. Soil, water, hospital environments. Generally, patients don't have Pseudomonas or Pseudomonas aeruginosa specifically as part of their microbiome. It tends to be an exogenous infection. It comes from outside the patient's own body. In hospital settings, they have found biofilms of Pseudomonas, so communities of Pseudomonas that are growing, replicating inside of soap dispensers. So as the physicians or nurses or your family is washing their hands, you're literally inoculating the hands with Pseudomonas. They grow pretty much anywhere. And I always used to get mad because this is one I worked with during my postdoc, because somebody would work in my staffhood with Pseudomonas and then Pseudomonas contaminated everything. And I always knew, because then my cultures were all blue. Why are my cultures blue? Who do I have to fight? And it was always some colonel that I had to go fight, because I did my postdoc with the Army. And they did not necessarily care for my rules. They had their own rules. They'll grow everywhere. So once Pseudomonas has taken hold in a hospital setting, it is always going to be in that hospital. There's pretty much nothing they can do to get rid of it. It grows in the pipes, it grows in the soap dispensers, it is everywhere. They have very simple growth requirements. I will still insist that they grew in pure filtered water. My lab mates said I was a little bit paranoid, but I'm pretty sure they did. They grow everywhere. They're aerobic though, okay? So these are not going to be associated generally with GI infection. So gram-negative rod from a patient sample, right? A wound sample, but not a GI sample. These are aerobic. They're also oxidase positive. So you look at a colony on a plate, maybe they didn't tell you what part of the body it came from on a patient. It's a gram-negative rod, but it's oxidase positive. Definitely not Enterobacteriaceae. They produce a number of different compounds that are involved in causing reactive oxygen species to form, to cause damage to host cells and to other bacteria. Pyoverdin they use to steal iron. Pyocyanin, so cyan, that's the compound that produces the blue-green color of the colonies. So it's a pigmented compound. It acts as a toxin to other cells, but that's why you see them as blue. And they have many, many virulence factors. This is a pretty dangerous bacterium. It is only biosafety level 2, but it's one that when you work with it in the lab, you may not see it, but I keep a pretty close eye on you, because it can make healthy people sick. So this one we don't play with. So it has a lot of factors to help it adhere. It has flagella, so it is motile. It has pili for adhesion. LPS can also be useful sometimes for adhesion, and it secretes carbohydrate compounds called alginate that can help it to adhere as well. It secretes a number of different toxins and enzymes. So it has a toxin called exotoxin A that disrupts host protein synthesis. Pyocyanin creates those reactive oxygen species to damage neighboring cells. Pyoverdin, they steal iron from either other bacteria or from the host. This is a culture that I grew in my lab at Fresno State. And you can spin down and see here's the cell pellet, and then this is all the supernatant, all the media, and it's just that blue-green color. So that's really indicative of Pseudomonas. It has other enzymes, kind of general enzymes that a lot of bacteria have, things like elastases, phospholipases, that help it mediate tissue destruction. And it produces another set of exoenzymes through its type 3 secretion system. So it has that same, remember we saw a picture of it in the Enterobacteriaceae on Monday, that like needle-like structure that it can make to inject compounds into the host cell. And so it can secrete enzymes that will lead to host cell death. And kind of why I was most interested in it is because it has high levels of antibiotic resistance. It's just like inherently resistant. It is inherently resistant, I believe, to either tetracycline or chloramphenicol. It's not even like it evolved resistance. It just is resistant. All of them are. But there are many multi-drug resistant strains now that are resistant to lots of different kinds of antibiotics. And so typically we see this as a nosocomial or hospital-acquired infection, or in our patients who are otherwise immune compromised. I would say that in my kind of experience talking about Pseudomonas as a clinical pathogen, we most commonly see it in burns, like burn wounds, and surgical wounds. And for whatever reason, there is something about a Pseudomonas burn wound that is almost like a death sentence. So when people get a Pseudomonas infection and they have, I'm not talking like, you burned your arm on the stove. I'm talking about, burns where we're talking about the percentage of your total body surface that was burned. So like 30% total body surface area with a burn, and you get an infection with Pseudomonas, that is pretty much a guarantee that your patient is not going to make it. It's a very, very dangerous infection. And they've done this study in mice where they've made like a surgical wound versus a burn wound of the same size, and they infected them with this bacteria. The surgical wound, fine. They get an infection, but the animal doesn't die. The burn wound, the animal will always die. So there's something about a burn that really compromises the overall health of the host that makes them really susceptible to these infections. So if you ever have patients that have burns, be very, very protective of them to make sure they don't get an infection, but especially this kind. We also see pulmonary infections, urinary tract infections. This one also is associated with causing swimmer's ear. If you've ever gotten like that external ear infection, especially after swimming, that's often Pseudomonas because it does well in water. Eye infections, if people have any sort of like damage to the eye, it can get in there and cause a pretty gnarly infection. One of my grad students used to, because I hate eyes, I think they're disgusting, and she used to always like put in her presentations a picture of a Pseudomonas infected eye. It's like all green and ugh. And she did it just to like, in the back like, woo, and that's why she did it. But also it will disseminate from other sites of infection and get in the rest of the body. So what we'll pick up with is unfortunately this slide on Monday, looking at a fatal infection and then talking about this case of folliculitis. So we will go upstairs now real quick and look at your plates that you haven't looked at already. We'll see if